Foundations of Mind group, we started due to a conference I organized here in March 6th and 7th. If you're interested in getting our mailings and so on, please look at the website foundationsofmind.org. And you can contact me through that. If you want to be part of our discussion group, send a CV. So that's foundationsofmind.org. Just look at the website, you'll find my email there. Thanks very much for coming. We have a real treat today. And I actually have to say that Walter has been so productive as an emeritus that his Wikipedia page is actually out of date already. And the Wikipedia page correctly says that he studied physics and math at MIT, which he may not care to admit to anymore. But there was a period doing electronics during World War II, which is deeply impressive. Philosophy at the University of Chicago, and then he graduated cum laude as an MD, being the third member of his family in linear ancestry to do that. His Wikipedia page says that he's published more than 450 articles and 4 books, and it's actually wrong. I counted at least 6 books. So here they are. So we have Neurodynamics and Reclaiming Cognition, the Primacy of Action, Intention, and Emotion, Society of Brains, How Brains Make Up Their Minds, Mass Action in the Nervous System, and most recently, last year with Quiroga, Imaging Brain using EEG. And the format for this is actually, Walter is going to give a demonstration, he's going to give his talk, and then he's going to answer questions for as long as people want to ask them, and as long as he wants to answer them, okay? The format we use is actually very, very open. You put up your hand, I call you to ask a question, which you then address to Walter. If you want to ask a follow-up, say you want to ask a follow-up. There's no censorship whatsoever, but we're actually really concerned on avoiding chaos. So this is our second Foundations of Mind workshop. The third one will be next week at the earlier time of 2 o'clock in the same place. So make a note of that. The third one will feature Carlos Montemayor. I'm speaking about philosophy. That will be at 2 o'clock in the beach room, same venue, on Friday. And that's the 9th of May. But I'm sure what we all want to do is to welcome Walter and hear him out.
Thank you. Okay, thank you very much. Thank you all for coming, and thank you, Sean, for making all this happen. So I'm grateful. I'm a biologist, and therefore have a view of consciousness and brain function, which is firmly based in experimental work, primarily in investigating the properties of brain images which the brain is continually creating in its efforts to survive and prevail. I don't know whether this is not as large as I had expected, but hope you can read that I'm talking about the evolution and neurodynamics of the action-perception cycle. And as a subtitle, I wrote when, where, and how consciousness and quantum field theory enter into the discourse. The fundamental element, then, of brain function is to direct the body into the world in search of information in order to achieve the goals that the animal and the person is continually creating in the course of its lifetime. And that cycle, the perception cycle, is most easily, I think, understood in terms of the basic topology of brain organization, how brains are put together into a functioning system. Next slide, please. Or am I supposed to do this?
Any key will do.
Any key. All right. Therefore, I look back in phylogenetic history of the development of the brain, which to a certain extent is recapitulated in embryology. And I came to the work of Charles Judson Herrick, a comparative neurologist of the 20th century, and his book, The Brain of the Tiger Salamander. And that's the animal that he selected as best representing in a surviving animal the primitive organization of the vertebrate brain, which emerged in the Ordovician sometime after 200 million years after the Cambrian explosion. And he wrote, "The tiger salamander is appropriately named, for within the obscurity of its contracted world, it is a predacious and voracious terror to all lesser inhabitants." That's beautiful Victorian writing, although it was published in the mid-century. So, without further ado, let's go to the object of his admiration. This is a view of the dorsal aspect of the brain of the tiger salamander, which is roughly 3 to 5 millimeters in anteroposterior extent, the right and left hemispheres. And what I've indicated is that this hemisphere can be divided, was divided by Herrick, into 3 main parts. An anterior sensory part, which was mostly olfactory. That is an important point because the explanation of how brains work is best couched in terms of how olfaction works, and enabled this animal to survive, to differentiate, to multiply, and come to dominate the biosphere. Then the second portion is the piriform cortex, essentially motor, comprising then the basis for the operation of the animal, both the musculature of the trunk for locomotion and primarily for the operation of the jaws in consuming food. This medial third is enigmatic. It's the precursor of the hippocampus, and its role is vital, as I will come to explain. Then we have here what he called a transitional area, TA. That essentially is where everything else comes in to the brain from the other senses, sight, sound, and touch. The operation of this, each of these hemispheres is locked together so that either one or the other can be studied and get the main dynamics. The action perception cycle, then, being a closed circle, is arbitrary where you decide to start. And I will state that it essentially is a pattern of activity which emerges over the whole of this surface in what we call a phase transition, that is, from a chaotic, disorganized, and low energy state or phase to a highly energetic state of coordinated, synchronized oscillation over the entire surface of the hemisphere, which persists for on the order of 1/10 of a second. That is a phase in which the motor action, previously proposed or developed or constructed for action into the environment has, through the environment, closed a loop to gather information by its action into the environment. And the sensory stimuli which fall onto its sensory surfaces, the ear, the eye, and particularly the nose and skin, that collection of patterns of input was in fact predicted by the animal in anticipation of its motion, because the patterns of activity are essentially the result of its action into the environment, and plus its prior preparation of what is to come next. Well, that's an overview then of the action perception cycle, which is the preparation for the collection of information, the action which brings discharges or brings stimuli to the collection of sensory input, sensory cortices, and then leads to adaptation, confirmation of the activity in the animal, updating the sensory systems and setting up in preparation for the decision of what to do next. Now, this is the primitive brain, and this is what happened when evolution took over, and this system then became elaborated through differentiation and natural selection into a broad range of species. I call your attention here to the brain of the whale versus the brain of the mouse. These differ 10,000-fold in their volume and therefore in linear distances within these organs. And it raises the evidence, presents the evidence, that one of the main characteristics of brain dynamics is that it's scale-free. That size is no barrier to the operation of these dynamical systems, with basically the same operations of the action perception cycle, very close in time, but differing within the organ by major differences in scale. But those make no difference. I call your attention also to regions in the basal brain, the cortex, of the entorhinal cortex, which plays a crucial role for this transitional area. What used to be the contributions coming to the TA now all come to the entorhinal cortex, which is the point of entry or the site of entry into the hippocampus. And that brings us to essentially the question of what exactly is happening in this window of observation, the action perception cycle. The time duration is on the order of 200 to 300 milliseconds. This is essentially the time segment which must contain the major operations of the action perception cycle. What you see here is the recording from the olfactory bulb and the measure of respiration, showing inhalation bringing about what we call a respiratory wave. It's in the theta range, at the crest of which the system breaks into oscillation. That's the manifestation of this phase transition that I've been talking about. And it is the basis for the depolarization, the excitation of the olfactory bulb, such that it goes unstable and breaks out into this oscillation. It is not so apparent that this increase in activity, in fact, has a characteristic increase in the density of the activity within the bulb, where from a state of sparse firing, irregular, unpatterned, and sporadic, there is a shift to a condensed state, as though this were going from a gas to a liquid, in which all of the neurons are directly participating in the oscillation, but with firing which is time multiplexed across the populations of each local area, each column of dendrites. In other words, the firing, looking at individual neurons, appears to be random, but in fact is phase-locked to the underlying subthreshold oscillation. That oscillation in itself, as we record it, is a field of electric potential, the electrocorticogram, that is epiphenomenal. It doesn't have any significant role to play. What it is the manifestation of the dendritic currents which are cooperating, collaborating, synchronized, and occasionally an action potential will form, and that now is an expression of the probability of firing. And when we look at the statistical properties of this probability of firing, we find that there is a pulse probability wave for each of the neurons involved in the transmission. And the probability essentially becomes a relative frequency over the population, and we can conceive and do observe with time averaging that the sum of the neurons expresses the activity at each point. That now is the observation that we make within this time window. Now, when the animal breathes in on a given cycle, it gets the information that it was looking for in a form which is an expression of the category that the stimulus belongs to. Let me present this in terms of a wiring diagram to show you what is happening. We have an array of receptors on the order of 100 million in each nostril for most vertebrates. And that number has within it on the order of 1,000 different types of receptor, genetically determined. That 1,000 types out of 100 million means that there's 100,000 of each type. And each inhalation brings excitation by an odorant, which will excite some small fraction on the order of 100 to 1,000 out of the 100,000. That creates an enormous problem for the olfactory system to have solved 300 to 400 million years ago of how can the animal generalize from this sample, which has never occurred before, will never occur again, and yet clearly belongs to a certain category. This essentially is done by formation of a Hebbian nerve cell assembly. I've schematized that by showing the excitation of two receptors feeding into a collection of 5, which has been formed by prior learning under reinforcement to connect to each other more strongly. The amount of increase in connection is rather modest, on the order of a fraction less than 1, or 1.2, 1.3. That increase in connection strength between these has a profound effect by the fact that it's cumulative over multiple sniffs. Not too many, 10, 15 is ample. That strengthening of the synapses pairwise with reinforcement over this set of input creates what we call a Hebbian assembly, after Donald Hebb, who first proposed that neurons that fire together wire together. That's Carla Schatz, who actually coined that fire and wire together. The result is that no matter which of the neurons feeding into this assembly is activated on any one trial, the entire assembly ignites. And this, we estimate, may contain on the order of 50 to 100,000 neurons wired together, and they fire vigorously. And physiologists have been observing these bursts of firing for several decades now. That dense firing increases the activity of that Hebbian assembly to a point where it leads the bulb into a state of sensitivity such that this condensed firing is ignited, meaning that the entire olfactory bulb goes into a pattern of oscillation, expressing now the category to which this odorant belongs. And notice that this category is only defined by dyadic pairs activated under reinforcement. And so it says nothing about what the chemical nature of the odorant is or its molecular structure, just that we have this coincidence of the firing along with the reinforcement. It's suitable for a primitive organism that doesn't have much else on its mind. This was anticipated by Lashley, who noted that generalization is one of those primitive, basic functions of organized nervous tissue. Here is the dilemma. Nerve impulses are transmitted through definite cell-to-cell connections, yet all behavior is determined by masses of excitation. The problem is universal in activities of the nervous system. So this is the solution to Lashley's dilemma, which was, in fact, at the time, right under his nose. This leads me now to the core of what I want to describe as the process by which 4 steps lead to the creation of knowledge and meaning from microscopic sensory information. And I list as the first step the abstraction and generalization of incoming information from the senses by the mesoscopic Hebbian assemblies, which now are oftentimes seen as concept cells, according to my co-author, Rodriguez Quiroga. We're talking now about a transformation at the input, the first synapse going into cortex, of microscopic, sparse firing carried by a topographically organized pattern of input into the olfactory bulb, where there is this creation of the Hebbian assembly. Now that Hebbian assembly we call mesoscopic because it has the fundamental difference that it is not a part of the external world. This generalization exists only in the brain of the animal and is a reflection of or manifestation of its learning and its modification for the particular of the environment in which it is living. So it is, in fact, an expression of mind, the primitive mind of the animal. It is essentially a property that exists nowhere else than in the individual. That's why we call it mesoscopic. Now the next step is the most difficult to understand, and that is the transformation of the phase, the state of the olfactory bulb from this low density, low energy, chaotic, disorganized state into the coherent oscillation. And that's what we conceive as the key operation to create the meaning of this stimulus, not merely its category. The category is critical for creating the specific sensitivity to an odor. So this can't happen just casually. It's enormously expensive energetically to give out the pattern of organization that constitutes the memory of this stimulus. Now, the memory is not merely some kind of pattern that is activated by the stimulus in the manner of retrieval of a memory in a computer. What it actually consists of is a set of instructions of how to create anew the recollection of prior experience with this odor in its portmanteau of meanings that it carries with respect to whatever it can identify. Now, most animals are equipped only with enough recall space, memory space, to identify anywhere from 5 to 10, 15 odors. That's all. Humans, of course, have an enormous capacity, but they have language. Or we have language, and that greatly expands our categorization. The basic olfactory system that I'm describing here now is far more limited. That being the case, then, we can go on from the phase transition, which gives the memory of the stimulus as I've defined it, and we go to the next step, which is the spatial delocalization. There is, in fact, a necessity in each of the sensory cortices to expunge the particulars of how a given stimulus is to be recognized and recalled, and its specific memory can be transmitted forward, freed from all of the detail from which it emerged. This is now dependent on the fact that there's a carrier frequency, a narrow band, typically in the gamma range, though sometimes beta, and that oscillation provides the basis for an extraction, a reduction of the information content in the output. This is because the activity which is summed by transmission through this output pathway is distributed across the spectrum. But now, as the summation proceeds over time, the different frequencies will cancel each other, leaving only that which has been adapted, has been created by the animal at the carrier frequency. Everything else tends to be washed away. This is the step of abstraction, in which not only is the particular information about which receptors were activated irrelevant, this process also now reduces the complexity of the transmitted information by transmitting only that which the animal has created. That is the step also that makes it possible for the different patterns, the different memories coming from the visual, auditory, and somatic cortices to combine together in the entorhinal cortex, because they all have the same code. And we simulate that very simply by expressing each memory as a vector from our 64 measurements of the amplitude at the surface. And that vector then can be expressed as a matrix, and we simply do matrix concatenation to simulate what the entorhinal cortex is doing up to the point of its own ignition. And then there's one further point, which is the sampling of this delocalized, transformed memory by the deep pyramidal cells in the piriform cortex. In effect, there is a reduction in the transmission to a comparatively small number of action potentials, on the order of 200,000 or 300,000, which transmits the integrated signal to the appropriate place for summation. This is now a change of state, first from microscopic firing of the sensory input to the phase transition to the delocalization, and now downsampling into another array of sparse firing. That sparse firing now contains the memory, as opposed to the sparse firing at the input, which has the sensation. So in a single step, with this phase transition, and now the downsampling from that phase transition into a sparse array of action potentials, has carried out a major step in which most of the information proceeding further in the output has been contributed by the bulb, not by the input. One of the things that people in sensory physiology substitution have learned is that there is an astonishingly small amount of information necessary coming through the eye or the ear or the skin or the nose. An astonishingly small amount of information can, within a small fraction of a second, bring out a combined memory locally and then at the entorhinal cortex globally of the situation that the animal is facing, including its memory from the past of what it has done about this in order to survive. So those 4 steps now comprise the operation which transforms the sensory information coming in from the nose into a perception, an olfactory memory. The next step is that our next stage, I should say, is the combination of this olfactory memory with the memory from all the other. And this refers back to our original picture of the forebrain, which essentially incorporates the hippocampal incorporates all of the senses in a transmission into the hippocampal formation and then back out to the entorhinal cortex. There's a third stage in which this same sequence, going from microscopic action potentials to microscopic action potentials through the condensed state, which leads now to activation of the entire forebrain, which is pretty obvious in the what do you call it, a salamander. And here I show a rabbit brain. Because of this scale-free property, we have the option of studying any of the array of vertebrates, whatever is most convenient. And certainly that justifies going to the rabbit or the cat or the monkey, which are intermediate between the mouse and the whale. I've shown here a diagram of the average size of these domains of activity in the neocortex, but I'll come to that later. What I'm emphasizing here is the olfaction feeding into the piriform cortex, and then buried within the brain is the entorhinal cortex, to which these windows feed. That is, these are the designation illustrations of our electrode arrays, which we've placed on the surface, showing that all the sensory cortexes operate the same way, and so does the entorhinal cortex. So now that establishes the window of entry, and here I illustrate a pattern of such an assembly from 64 electrodes forming a window onto the surface. And here you can see an example of the systematic single frequency that transmits a signal by virtue of the pattern of amplitude modulation. And these are contour plots of the amplitude, the average amplitude root mean square of the activity of the carrier wave at each of 64 points, with highs versus lows done by contour plots. This is an illustration of the change from control air to an odorant. We give significance by reinforcement, showing the change in pattern. Now that made us very happy until the next couple of weeks. We watched this again, looking for these same patterns, and found that they were not the same patterns. And that led us really to the amazing insight that these are not representations of stimuli on the surface of the skin or the nose. These are registration of something created within the animal, and namely its memory. And that can help us explain how these continue to evolve. There is still a difference, but it's more complex and difficult to see. But nonetheless, we know now that we are not looking at representations of stimuli. We're looking at the animal's record of the meaning of the stimuli. We do this by expressing each as a vector, each pattern, 64 amplitudes, and that defines a cloud of points in 64 space, which we project down into two-space using stepwise discriminant analysis to show that here is a control with a pattern mean, the geometric mean for the background, and here is another cluster which is a manifestation or the showing of the basis for characterizing the difference between control and reinforced odor. And here is an unreinforced odor which we require the animal to identify. It gives us a third cluster. The memory of the categories is based in the classification using for each sample, which of these centers of gravity is it closest to the control, to the reinforced, to the unreinforced. What is significant about this illustration is the fact that the classificatory information is uniformly distributed over the entire olfactory system. These are illustrations that we can get the best classification by using all available data from all parts of our window of observation. If we remove any of these randomly selected over multiple trials, we find that the goodness of classification is systematically reduced with the extent of removal of contributions down to the minimum here of 8 electrodes. This shows us now that the location of the information for classification is distributed. It is mimicked easily by a Fourier transform and therefore conforms to Carl Pribram's notion of a hologram only in respect to the capacity for delocalization. The memory is distributed throughout the coherent oscillation and is nowhere pinpointed. In fact, that gives the property that the transmission from the bulb goes out to an indefinitely large portion of the forebrain, and the transmission to each local part is the same. The same information is sent out at reduced resolution in the manner that Carl Pribram has used to demonstrate by taking the condenser out of the projector and showing that the information is everywhere. The same information at whatever resolution is needed by the brain. So there's no loss of information. Where it stops being a hologram is that holograms simply hold information, whatever is put into it, in this non-local form, and never make decisions. They don't throw away information. They save everything. And it's up to the readout of the transmission to carry out something in the nature of pruning, of reducing the total information to just that which is needed locally. Now, I emphasize that there are these 3 stages. First, from stimulus to percept, next from percept to concept, and the next, the global transformation to complete the action perception cycle by bringing the next action into play. This is in the gamma range primarily. In the beta range, we have this kind of formation of a high energy, high density state repeatedly, only relating to the stimuli in this narrow time strip between the onset of the conditioned stimulus and the onset of the conditioned response. In our case, a respiratory reflex. Now, let me pass by this, it's too complicated. I've covered the abstraction and generalization and the recall of memory. Now I want to address again the question of delocalization by showing that there is, coming in from the receptors to the olfactory bulb, this topographic mapping, as in every other sensory system, but showing now that the next step is this delocalization, where you have from each local part of the bulb, there is a broad distribution to the olfactory cortex, and conversely, that to each point in the olfactory cortex, there is a broad distribution of input. These patterns now have only recently become widely recognized by the use of a new technique, diffusion tensor imaging, in which the broad distributions of input output have been clearly documented. In the case of the olfactory system, I had to do this with microelectrodes of systematically stimulating each part of the bulb and demonstrating that I activated the cortex broadly and conversely stimulating in the cortex, antidromically activated a broad swatch in the bulb. So this simply nails it down that something that's been right under our noses all this time are these global transforms which operate so quickly to bring about the activation now of the entire hippocampus through the entorhinal cortex located in here, but in the primitive bulb the interactions are in a continuum. This is another feature that's not commonly recognized, that the neocortex in mammals is a single organ. There are no glial barriers from one part to another. There are certainly local differences, highly important for the pre-processing of sensory information, but owing to the fact that the brain dynamics is scale-free, when a transformation is initiated at any point successfully by a stimulus that has passed the initial screening test, that goes to the entire neocortex and it goes into coherent oscillation. So looking at it from the standpoint of the recordings from humans of these dendritic fields of potential, we make a distinction between the electric field of the encephalogram, which is epiphenomenal. These are the small, minuscule microvolt potentials that are created by the neurons actively participating, actively processing information with a current loop. Loop currents mean that whenever a neuron fires or has a dendritic wave of potential, that doesn't create or destroy charge. It implies a loop of current. Current is moved from one place to another, like batteries. And the excitatory currents essentially have an internal IR drop at the membranes where the ionic currents are crossing out or in, outwardly in excitation, and on the return current path they go through the extracellular space, low resistance, and that's where we see the electrical potentials. The same current that generates or that controls the firing of neurons, but this gives us a direct access which gives us insight into how the system works. So this is now recording from a standard array of 64 electrodes as spaced at the usual 2.5 centimeters to 3 centimeters spacing on the surface of the scalp. Much too coarse for fine-grained analysis, but it's convenient to have that. And this shows some illustrations of the pattern of excitation for control and for a reinforcement odor. And it's difficult to see the differences between the amplitude pattern here and here. This is the original, and this is re-referencing. There's a great problem, technical problem, of identifying patterns in the scalp, which requires some pretty sophisticated potentials, but nonetheless, it's feasible. It's done repeatedly. And what we find now, again, is the probability of there being a difference between the control and the reinforced stimulus presentation is that there are 3 episodes of statistical significance between the CS and the CR, the conditioned stimulus and the conditioned response. And the third one is the one of greatest interest because that covers the time period where the animal now, or the human in this case, is getting the results of the passage of the excitation, the recall of memory from all the systems combined into the gestalt. It's gone through the hippocampus, come back out to the entorhinal, and now the entorhinal is sending the sparse firings to all cortices, both motor and sensory, to initiate the next response, to decide what to do and what to expect. Now that is essentially the basis, in my view, for the awareness of the individual, whether animal or human, the awareness of the impending action about to be taken and of the expectation of what is going to happen when the action takes place and provokes discharges into all of the sensory cortices from the change in the relationship between the subject and the environment. So we have the existence of this prior awareness that action is about to take place, and then the action which brings about the changes in the sensory system, bringing it through the primary sensory cortices, bringing it to the entorhinal cortex for fusion, and then bringing it back out to the entorhinal cortex, but this time with a label applied by the hippocampus of where the subject was at the time this event took place and what time it was. When did this happen? So that's my picture of the organization of the activity of this primitive form of the olfactory, of the vertebrate brain, which is dominated by olfaction, but which provided the model by which all other sensory systems became organized themselves around this primitive state. So, a primitive thing. And this has the same property of delocalization. No part of the cortex under the scalp has any greater or lesser information than any other. It's truly a distributed system. And so my old teacher, Carl Pribram, read my thesis and allowed me to graduate 60 years ago. That essentially is the basis for my claim that this phase transition, particularly this third one, enabled by the fact that their dynamics is scale-free, this third one means that the entire cortex is sustaining this brief period of fluctuation in the beta range, and that is the basis for our awareness that this is happening, because it's a real challenge to think of any better way for us as individuals and for animals as well to have access to this immense treasure of the unconscious as a resource for what to do next. Now, that essentially leads me back to these 4 steps, which are repeated in each stage of going through abstraction and generalization of the microscopic action potentials coming in to the destabilization, the activation of a phase transition to a high density of firing and of activation, and then to the delocalization of this event, followed by down sampling. And my proposal is that this sequence, this at each of the 3 stages, basically the same, that is the primitive algorithm which enabled the vertebrates 400, 300 million years ago to flourish above all other species. So that is my message about the olfactory cortex, and my concluding hypothesis is that what distinguishes knowledge from information is the condensed state of the global cortical neuropil, in which every neuron within the coherent domain in a wave packet coordinates its learned dynamics, working together only on the basis of past experience. I list the tools needed for this as nonlinear neurodynamics, multivariate statistics, far from equilibrium thermodynamics. We've got a nice development using the Carnot cycle to define our variables. In random graph theory, neuropercolation, and of course many-body physics or quantum field theory, according to my collaborator. And these are the tools that I propose. Non-linear thermodynamics. We can read about all this that I've talked about here in a book with Kian Quiroga entitled Imaging Brain Function with EEG, published just last year. And there's more material about generalized extreme value distributions by Rice, and of course the most recent work on nonlinear thermodynamics. So I had one alternate title of this, which I'll share with you, that my picture is that consciousness emerged sometime in the last 500,000 years as the plan that hunters developed of what to do next when faced with obstacles in the field. And that's, I think, the real basis for bringing out consciousness into the form that we experience. So thank you for your undivided attention. I'm much appreciated. Thank you.
Thank you so much. I just wanted to remind you what the format is from now on. We're going to have a Q&A session. Walter's kindly agreed to answer questions. Please put up your hand if you want to ask a question. Address your question to Walter, and then announce if you're going to do a follow-up. And we're not going to have another presentation today. I don't think any of us really want to follow Walter. And so please, let's let's have a Q&A right now. Before we do that, I just want to thank a few people. Justin Riddle has helped us a lot internally, booking the room and so on. We had Zoe help with the setup at the start of this. The tech guy was not here today. And I really would like to just thank Justin and Zoe. Okay, the floor is open. Please.
Great presentation, thank you. Can you elaborate a little bit on the role that deep pyramidal cells play? I saw that it was in step 3 of your on your 4-part plan there, and I was just confused about what role the deep pyramidal cells exactly function in the system.
Middle cells?
Pyramidal.
Oh, pyramidal cells, yeah.
Ah, sorry.
Yeah, this in particular, the projection neuron in the olfactory bulb is called the mitral cell, but it has all the characteristics of a transmitting cell. In the olfactory cortex to which this is sent, there is an additional layer. At the top is the superficial pyramidal cell layer, and they're the ones that are generating the electric currents that we record with the EEG. Now there's a deep layer of pyramidal cells, deep pyramidal cells, which are larger and which have a larger radius of integration. They're the ones that are downsampling the pattern at the surface, recorded at the surface, which is a manifestation of the activity of the superficial pyramidal cells. And those we can readily show are related to the field potential, that is to say, these dendritic currents that we can so easily record, which I've shown illustrations of. They are the fundamental manifestation of the distributed character of the memory that has been transmitted from the bulb. Now, that's what is being downsampled by the pyramidal, the deep pyramidal cells, because they are the ones that have the axons that carry the transmission to the hippocampus, the entorhinal cortex, and then to a number of other basal structures. But only a portion goes to the the central hypothalamic projections. Those are all essentially carrying the same message, but at reduced resolution. And presumably by evolution, the entorhinal cortex will always get enough of what it needs in order to form its contribution to the gestalt. Similarly, with all the other cortices, they are doing a similar projection. We have not identified which locations are performing the operation that we've demonstrated for the piriform cortex. And we think that diffusion tensor analysis will give the necessary anatomical information of where to look and what to record from. Yeah?
I'm curious about how we can reconcile the types of activity we see in mesoscopic patterns with the types of activity we see in single cells. So for example, many people, including your colleague Count Quiroga, look at the receptive fields of single neurons, and for example, in a place like the face area, you can find cells that seem to be largely silent and then spike vigorously in response to a certain face.
Mm-hmm.
So how do we take the seemingly feature-selective behavior and reconcile it with this idea of a holographic representation that presumably dissolves a lot of the sensory information by the time it gets to an area as abstract as the face area?
Well, I think that the key to any resolution of this problem is to find correlate at either of two levels. in one respect, for example, if we're recording in the entorhinal cortex, we can see some that do fire vigorously, which constitute the Jennifer Aniston cell and related types. I think those are manifestations of Hebbian assemblies. That is, these are learned, obviously, because it's only in individuals who have been exposed to Jennifer Aniston that will remember her. I'm sure that I wouldn't have any. I have no idea what she looks like. But that would be to find a nerve cell assembly to identify it doesn't have, or unlikely to have, modulation according to the carrier frequency, because it's selecting a target of a cell assembly of a basin of attraction. Now, that is on the outflow to the hippocampus, and it's essential to have that throughput to the hippocampus in order to attach the memory of where you saw Jennifer Aniston and when, in order to make that a permanent part. This is Larry Squires years ago showed that the passage through the hippocampus is necessary for a laying down. But coming back out again, you have another opportunity for a coordinated firing, but in this case, it's the initiation of the transmission from the entorhinal cortex to the hippocampus. My postulate is that you have two steps. One is the activation of the Hebbian assembly, which was formed by learning, which triggers the formation of a wave packet, in my terminology, in the entorhinal cortex for transmission of the multimodal sense wave packet. Well, that means that you have to combine recording with local field potential to verify that there is such a wave packet at the time you're recording. And according to our theory, our construction, that doesn't show up as a vigorous burst of firing. It shows up as a coordinated, cooperative firing with numerous. Now, whether you can get numerous cells, I mean, how many do you need? We can go up to maybe a few dozen in one small area, like the entorhinal cortex. But if you try to put in too many electrodes, well, you've had enough experience to know that you can't put indefinitely large numbers of electrodes in without destroying what you're trying to record. And so to make this distinction between the mesoscopic, which is the assembly, and the macroscopic, which is a statistical phenomenon requiring hundreds, if not thousands, to define the local variable. In fact, I would say that our experience is that when you count the number of pulses that are needed to form a statistical ensemble sample to show the macroscopic participation, you need on the order of 10,000 pulses. And to do that, the neuron that's firing at 1 per second or 2 or 10 per second, it takes a long time. So then you run into the problem of time variance. But it's a fascinating question, but those are the issues that we have to deal with in order to proceed further. Yeah.
Very nice talk. One of the interesting things that you had found were for the macroscopic stage, where you showed the topo map of the EEG, but then over time the topo maps change.
Yeah.
So at that level, where might be the information?
Oh, it's global.
But it keeps changing?
Yeah, yeah. But what happens is a change in the location of the point in 64 space.
Yep.
And that now means that in any one day recording session, there's enough stability to show clustering of repeated presentations of the same stimulus. They're not identical. There's always this, as I showed, variance, but nonetheless they will categorize. And that's our major evidence, is the categories of images. That's why I'm so proud of this book on spatial imaging of EEG, showing you how brains work. You wouldn't have any access to the really interesting macrodynamics without the access to the dendritic potentials. And most people, I think, have mistakenly concluded that because they have the same frequency, the carrier frequency, that it's just volume conduction, just overlap of potential, trivial. Well, if you look at the amplitude modulation, you find there's serious differences with electrodes that are very close together. They have the same correlation coefficient because they have the same frequency, but they're very different in respect to the phase and amplitude.
I will buy the book.
Yeah, I recommend it highly. Yes?
John? So the wave packet, that's an extracellular potential? It's a combination?
Well, it has the wave packet has its manifestation in the local field potential and the EEG. Yeah, so you can record it even from the scalp.
So how is that read? I mean, it's basically, you're saying that's a downsample version, and then that's read by the interrhinal. Is that what it is?
Yeah.
And so I don't know enough about neurobiology.
Oh, okay.
Yeah.
Well, look at it this
because
way.
It seems like such a diffuse thing.
Right. Well, the interrhinal cortex is a very definite structure, and in fact, it's been recognized now as neither archicortex or neocortex. It's mesocortex. So that's a designation simply that says, hey, this is different, but it's important. And it's the portal from all of the sensory systems into the hippocampus. Now, this means that the entorhinal cortex now is a focus, is a central component for brain function, not the thalamus. Most people are concerned with the thalamocortical relationships and are neglecting the entorhinal cortex, which from the standpoint of the action perception cycle is the crucial nexus coming together from all of the senses and then making a construct which contains all of it, making a wave packet.
I have a follow-up.
Yeah.
I'm curious, but more specifically, because I don't really know neurobiology, but I usually think of it axon and dendrites and
That's what most people do, yeah.
But now you're talking about a
population.
You're talking about a population.
Yeah,
So
that's the title of my first book, Mass Action in the Nervous System. That's on my website,
But
too.
is population expressed through a diffuse potential? Is that what you're saying?
Yeah, well, it's expressed in two ways. One is the coordinated output of all of the axons coming from this population. That's the real vector of effect. And then the noise that it makes.
I see. So the actual potential is sort of just an epiphenomenon. It's the actual...
Well, the action potentials, yeah, they have their own epiphenomenal pulses, but
I'll stop there. I'm confused. I'm just getting one.
Well, it's the pulses that are the output. The action potentials coming from the neurons, the transmission neuron. That's the real output, and that is a vector.
Sure.
The young lady at the back, Walter.
Oh, yeah, please.
Somewhat methodological question. You mentioned volume conductance problems with scalp EEG in humans, and I'm very interested in features that are shared globally, and also locking of the phase, alignment of phase within a specific band across distant regions. What's, in your opinion, a good way to go about this? I mean, current source density removes a lot of the signal. Imaginary coherence, imaginary phase locking, it's suggestions that people have made, but all of this removes a lot of the signal. So what's your suggestion if we want to look at that global level of information? but have only access to Scotty EG and are fighting with conductance.
My suggestion is you invite me for lunch. Please do. I mean, it's a very, you're asking into technical details, which I've tried to minimize, recognizing that, I'm not speaking to neurophysiologists and trying to persuade them of anything. But, I would like to come up to me afterwards and we'll discuss that. Mm-hmm. Other questions?
Carla?
I'm hard of hearing.
I have two questions. And one of them is associated to a particular diagram that you showed of going through a phase transition to an oscillatory motion of the olfactory system. And I just was curious whether that has been categorized as a first order or a second order phase transition, whether it's continuous or discontinuous. Has there been any categorization?
I think I would prefer to delegate that question to a physicist, since I think that we're somewhere in new territory of applying physics to places that it wasn't designed for. Now, the fact that we have these abrupt discontinuities in the onset of the burst of oscillation, my wave packet, where the phase literally jumps within one digitizing step to the next to some arbitrary point on the cycle of a new frequency, suggests that, this is a first order phase transition. But the end of the burst of oscillation is equally important. How does the thing stop? How do you get it out? Well, that's a second order type phase transition because it's more gradual. And we use that, use the mathematics of extreme distributions developed by Stephen Rice 60 years ago, 70 perhaps, where the carrier frequency is not just one frequency, it's a narrow distribution. And the width of the distribution will determine how soon this system will go out of phase and collapse because of the dissipation of energy by the frittering away with the cancellation of the neurons that are now working against one another because of the shifts in phase. The interesting part of his theory of noise is that when you have this background, which we always have, of random activity, well described as scale-free, that when a narrow band frequency is imposed, which is done by the cell assembly, and that creates a frequency spike in the spectrum, which then will broaden with time as they're going out of phase and reach a critical level at which the system will collapse back into chaos. So that's the best answer that I can give now, that it starts with a first order transition and then goes with a second. And the time duration is fixed by the mean frequency of the displacement, so that there is a built-in termination of this wave packet that will get rid of it, because it's important that an animal essentially within 0.2 of a second be ready to take another action perception cycle, and it can't do that until it's wiped the slate clean.
Yeah, sure, of course.
So the second question is associated to the non-local interaction that you mentioned. So it looks like that there is some non-local global action in the entire section of the olfactory brain that you showed. So my question is, when usually we talk about non-local interaction, there's usually some sort of a mean field
Sorry, I was distracted. Maybe you can repeat the question for me.
Please, Carla, if you could. Mean field, please. Yeah.
So usually when one talks about some local interaction, there's some sort of a mean field, and then there's some, wave function, for example, that couples to that mean field. So in this case, how would you map this sort of categories information in some sort of a wave pocket, and it's actually coupling to some neurodynamical mean field explicitly said?
Did you get it?
I think we've got an issue about non-local. It's a kind of allosteric question. We've got some action at a distance occurring, and there has to be some field whereby this action is transmitted. A mean field. This is what you question. And yeah.
They can also talk to you about it.
Well, I can make an effort to answer. I'm not sure what the question is, but put it this way. I think mean field statistics is not applicable here because what is important is the preservation of the information in the neuropill, the microscopic neuropill, such that the memory itself is an extremely rich mix of a great variety of past experiences pooled into this one global discharge, this one global wave packet which covers the entire cortex. And that distributed information from past experience is what makes the difference between a mere accumulation of information and the knowledge and meaning. So this is where I disagree with Tononi, who has piles of information but doesn't have the mechanism by which it is assembled. So that's one part of the question. That's redressing the mean field, that is not a mean field. And the second part is, how does the great extent of this coordination, this synchronization, form over the entire cerebrum within a quarter of a cycle of the carrier frequency? And that is done because the connection between neurons is governed by a distribution in which the log of the density decreases with the log of distance. So there's a great number of very short connections, mainly interactions with water dipoles, and a very small number of connections with long axons which go the full extent of the cerebrum. These frontocortical or fronto-occipital connections being the best example. Well, it's those distributed connections in a log-log form which are scale-free, and that's what enables the phase transition to occur over the entire neocortex. That's the astonishing part. We have now such clear evidence for the formation of these scalp EEG patterns, which are notoriously widely dispersed in their origins, but which do have this property for what amounts to an instantaneous change. Because with a 50 hertz signal, this means now that within 5 milliseconds, the onset of this oscillation has begun over the entire cerebrum, irrespective of where the site of nucleation is, the point of origin of the distribution. So that's an excellent question. I'm not sure it's the one you asked, but that will do.
Okay.
Oh, wait.
One last question, and then we're going to end. Sperry, please.
Thank you. Thank you so much for your talk.
I can get a little closer to you.
I wanted to ask you if you would please go over your diagram, if you will, verbally or otherwise, the awareness itself. How does it remember and understand how to project a possible future as a dynamic, as a function? I understand all these ingredients that you've shared and pointed to are all dynamically working together, but could you describe awareness itself?
No. In a word, this is essentially research that I would hope people will now undertake, because we have shown that these global patterns exist over the entire scalp, but at low density. That is, the spacing of these electrodes is the standard clinical array with spacings of 3 to 5 centimeters between the sites of recording. But we know from spatial spectral analysis with linear arrays of electrodes, only 64 because that's what we had, that the optimum spacing between electrodes is 3 to 5 millimeters, tenfold closer than is commonly the case for the scalp. Now, that seems to fly in the face of everybody, but I think it's because people are looking at correlation coefficients, and they're extremely suspicious of the effects of monopolar recording, meaning referential leads, introducing essentially zero lag. Well, that's easily disposed of. But the fact remains that there isn't sufficient information in the literature to address the question that you're raising, which I think is crucial. Not only how do motor cortices organize the projected action, but simultaneously how does each sensory cortex prepare its pre-afferent message to tune the cortex to what's going to happen next. I haven't emphasized enough, I think, the fact that the incoming pattern, which is now categorized, has no meaning until it has a comparison with that preceding in order to project what's the next. And the animal, to do that, needs to save it and save the previous one. And that's what the hippocampus is doing. And then the animal needs to know, where was it when it took this step? Where is it now? And where would it want to be? Spatial localization. That's, go and hear Agarwal's lectures. You'll find out how the hippocampus does that. And similarly, you need to specify, when did it happen? And when are you now in order to take action? But more than that, you have to know, did the action that you intended actually take place? Did the animal move? Or did you move? That takes input from the somatosensory. And then, of course, you have to multiply the fact that this is happening in every sensory cortex. So this is now just one part of one action perception cycle. And all this has to be taking place within a few milliseconds. And there's no other system of analysis that I know of that can give an answer of that degree of complexity of how can all this get got together into a field. And when that field goes out, it involves all of the cortices, and I view this as a kind of superfluid channel for sharing information over the entire extent during this duration. And that this duration of this third integrated form is a definition of William James' psychological now. That they're all contained in this message, which is the whole cortex. How could that not be something that you're aware of, conscious of? I see it as ipso facto. that you have so many neurons which are all doing the same dance together? How could you not be conscious of it? Well, after that is a philosophical question. So thanks for your question.
Okay.
Thank you. Good to see you again. Good to see you. Thank you. Thank you all.
So just a few brief announcements. First of all, the Foundations of Mind is just an informal group. There is nobody behind us. There is no agenda here. It's just a group of people who've got together to discuss the kind of issues you heard today, particularly the new language you heard today about dynamics, phase transitions, and the idea of fields in the brain. This is exactly the kind of language I think we need to move towards. That's our website, foundationsofmind.org. If you want to get in contact with us, you'll find an email address there.
Thank you.
I thank you all for coming. We're here at 2 o'clock next Friday, 9th May at 2 p.m. 2 p.m. Friday, 9th May. And that's same place. I hope that you can make it. Again, I want to thank Justin Riddle. I want to thank Walter Freeman. I want to thank Zoe Zhu, who helped us set up. And I want to thank you all for coming. Hope to see you again.